Hi guys, so lecture 4. So we start to talk about HIV. Before that, we talk about virus in general. So therefore, viruses are not alive. So the virus has different gadgets, if you will. So there are the DNA inside and protein that's called nucleic acid, and around it it has capsid, and some fancy virus has phospholipid and others has spike protein. So the most basic virus has capsid and nucleic acid. So DNA inside and the cover outside. That makes a basic virus. And there are 4 ways of virus entering human cell. So because virus is not alive, so how does it infect us is through using our own cell to produce virus protein and then make copies of themselves. There are 4 ways. So one, endocytosis is just like engulfing the virus into the cell. And membrane fusion is on the membrane of the virus, there's like keys. And then on the membrane of the cell, they are able to connect and then recognize virus as a friend, so they let them in by both first the key connects and then the membrane of the capsid opens up and inside the DNA flows into the cell, human cell. Genetic injection. So you remember if you remember there's like a spider shaped virus, they stand on the membrane of the human cell and then inject their DNA like a syringe into the cell. And fourth type is simple, like without any channel or anything, it just goes into the cell as a whole virus. Primary, so moving on, there's a primary immune deficiency, secondary. So the key point to how to differentiate primary, secondary is primary is there is something wrong to the immune system directly. For example, this complement system, the cascade, has problem to be activated. The protein is missing or something else. I don't remember these two. You can look it up. But the key point is how to differentiate primary with secondary. Secondary is other illnesses or situation that cause deficiency in immune system like HIV, malnutrition. These don't focus too much on the terms or the meaning of the illnesses. Now HIV. This is what HIV contains. 2 single-stranded RNA, 3 different enzyme, and 2 layers of capsid and 1 envelope. So this process, I better show you a picture. Oops, no, not YouTube. The SCID. This kid doesn't have B or T cell production. DGS, disorder of the T cell. Here. Let me get a summary here. So first, there is the connection at the membrane. The CD4 connects with the receptor of GP120. Don't try to memorize the name of the receptor. Just knows that the virus receptor connects with the T cells receptor, CD4 receptor. And then the T cell thinks, okay, it's a friend. So they open up the membrane and then the envelope of the HIV opens up. So the double layered capsid and the enzyme along with the two single strand RNA goes into the cell. And then the capsid opens up. We have the RNA and the enzymes. Now the first enzyme called reverse transcriptase will transcribe this viral RNA into something readable by our own cell. And then we need the second enzyme integrase to integrate this transcribed our DNA into the host DNA. And then after it's integrated, we use mRNA. It's like the normal process of producing protein. mRNA read the portion of this provirus. Provirus is just a name for this new strand of DNA. Like partial is human, partial is viral. And mRNA brings a strand of this provirus into outside of the nucleus to the ribosome, and then ribosome will produce a protein, but this protein is virus protein. Now everything is assembled at the edge of the membrane, the cell membrane, and then it's coming out. Budding happens. Budding is like the envelope. On the envelope, they have proteins, right? So budding just refers to the protein starts to form at the edge of the membrane, and then this new virus is formed. However, inside of this virus, it's like a mess. The protein is made. Everything is assembled. Here, however, it's not cut. It's not, the material is there, but the strand of the RNA or the capsid is not formed yet. We need to use the third enzyme that's called, what is it? Where is it? So we need the protease. After budding happens, inside of the virus is a mess. So we need protease to cut the protein into order. The two single strand RNA double layered capsid. Okay, that's the process of virus, HIV virus. First, the membrane fusion. So binding of the receptor, and then membrane fusion, uncoding, then reverse transcription. The sRNA is transcribed into virus DNA, and viral DNA is integrated into the human DNA, and then mRNA comes, transcribe, like take a portion of that provirus, and then provirus that strand of is taken by mRNA. mRNA sends it to ribosome produce viral protein. When protein is assembled at the edge of the human cell membrane, budding happens and then it flows out. Then protease, the last enzyme, cut these protein into order and make a new virus. It's the process of HIV virus infecting a CD4 T cell. After knowing this process, so you are able to apply, we have 3 different enzymes. So there are 3 different medications that will stop different different function, just stop the job of these enzymes. First, stop one medication can stop the transcription of the virus RNA into DNA. Another medication can stop integration of the viral DNA into the human DNA. The third one is able to stop the protease forming the functional HIV virus. Okay. Let me check the presentation. So here's the drug. Reverse transcriptase inhibitors. I covered these, so you just need to take a look at the name of the medication. So the function of this medication is in the name, integrase strand transfer inhibitor. It's inhibiting the enzyme, the integrase enzyme. Integrase is used to integrate the virus DNA into the human DNA. So everything is in the name. Don't try to memorize it. Now, acute HIV is like high viral load 2-4 weeks after infection. Chronic is low viral load because patient has received the treatment. AIDS is when there's high level of viral load and usually patients die from opportunistic infection. There will be low CD4 T cells. There is no cure, only maintain viral load low. All right, let's do lecture 5. 555 vaccine. So you know that my notes are very short is because I just eliminate everything that is repetitively mentioned in nursing, and I think everybody knows. We don't need to waste time to talk about how important antibody is. Taking the antibiotic is remain to finish the antibody, etc. So lecture 5, it's about vaccine. Why giving vaccine is beneficial is because with vaccine we don't get infected, and then we don't use antibiotic, and then because we don't use, there's no need for antibiotic, we won't develop drug resistance. So next time if we really need antibiotic, it will work. So that's that. 4 types of the immune system, how you require immunity. 1, natural, passive. So when we talk about natural is we require it from outside. So, and passive means we received it passively. the antibody is given to us passively. For example, breastfeeding. Natural active is, yes, the antibody is from, we required it from outside, acquired it from outside, but it's actively acquiring, means we got sick ourselves and then naturally we developed the antibody. Next, artificial passive. So again, passive is the antibody is given passively. Artificial, because it's man-made, so we are receiving the antibody passively. Artificial active, so that's vaccine. So here are different types of vaccines. Okay, it's short. I just break it down. The main point of this lecture is different types of vaccine, and I group them into pairs so to help you understand and you don't need to memorize. So first pair, inactive and attenuated. So don't be scared by the name. We always scared by the name. Actually, it's just saying a dead virus and a alive virus. That's why I put it into a pair. These are two different types of vaccine. One is dead. We are giving the dead virus or bacteria to a patient. And this one attenuated is we're giving the alive virus. Something I need to mention for formaldehyde, I wrote it here, is because when it's the chemical liquid that we put the dead virus in to store it. As just a side note, not important. But here, this serial passage is important to help you. If we give this a live virus, you might ask, okay, isn't that if it's dangerous to give a live virus to a person, that person may die from this virus. So no, because we are giving a live virus, but this virus is modified in the lab through a process called serial passage. So serial passage is when we get the virus that originally will kill people. It targets people's cells and then give it to animals, like for example, monkeys. And through several generations of monkeys, and then we take that virus, this virus is modified. It attacks more the monkey cells than human cell. And then we give this monkey targeting cell targeting virus to chickens. And after several generations of chickens, this virus is more modified. It's further away from attacking human. Then we give the final product to human. The human will develop an antibody against this chicken attacking virus. But because they are from this, compare the original and the chicken attacking virus, it's from the same family. So the antibody is going to work on the actual deadly virus. And moving on to next one. Okay. Okay. Next pair is toxoid and subunit. They are both part of the bacteria or virus. Toxoid, it's in the name, the meaning. It's using the toxin, but again, it's not actually the toxin, it's a denatured protein that has similar appearance of the toxin. That's why it's harmless. And because it's only like it's harmless and it's denatured protein, it's not going to have a strong memory of this virus or bacteria. So this vaccine lasts for a while but requires adjuvant to boost it up. And we require several doses. So an example of toxoid vaccine is tetanus. Next, the subunit. We are using parts of the virus or bacteria to develop immune response, develop antibody. For example, the envelope, or it uses the polysaccharide, use the outer layer, the peptidoglycan, for example, because that is made by carb, like polysaccharide, it means carbohydrate. But here you have to know that carbohydrate is poor at inducing immune system, means it's hard to develop an antibody that react to this specific bacteria or virus really quickly. It's hard because this vaccine is made by carbohydrate. We only have few types of carbohydrate in our brain compared to protein. Protein induce strong immune system response. And that's why when we use protein to use as vaccine, the antibody that develops at the end is able to detect the virus really quickly. And in this case, if it's polysaccharide, a carbohydrate, it is harder to react to the virus once it enters. But why do we use carbohydrate if it doesn't induce a stronger immune system? It's because some bacteria has a thick layer, remember, or a virus maybe has a layer of a carbohydrate outside of their body, outside of the cell. So it's because it's a most outer layer, we have to detect the carbohydrate first in order for the more white blood cell to come and induce immune response. Here I said an example of a thick polysaccharide will be Streptococcus pneumonia. Next is conjugate. Okay, so the toxoid is using adjuvant as a boost. And here we have conjugate to boost the carbohydrate. So because carbohydrate is poor at inducing immune system, we add protein to it. So that makes a conjugated vaccine. Yep, that's it of what you need to know. So moving on to the last pair of vaccine, RNA, DNA. So these concepts are similar. The difference is just we use either viral RNA or we use viral DNA. If it's RNA, then that makes this vaccine RNA vaccine. If it's a viral DNA, then it's a DNA vaccine. How does the pathway goes? It's let's say we use a RNA, viral RNA as a vaccine. We inject this RNA into the cell, into our body, and then it uses our human cell to produce viral protein, just like HIV. What makes it brilliant is that now we are able to use our own immune system against this virus. You may ask, okay, then isn't our human cell is going to make a live virus that's going to attack us? Then we cannot give it to immune compromised patients? The answer is no. The fact is it's safe to give immune compromised patients. Why? Because the strand of RNA we give as vaccine only enables the our ribosome to make a capsid of the virus, only parts of the virus. That makes the virus that our own cell produce harmless. So that's why this type of vaccine is brilliant. All right. Oh, just if if we are using a strand of viral DNA, it will be first transcribed into RNA and then goes to ribosome and then produce protein. Next, last concept in this lecture is the herd immunity. Herd immunity is in a in a community, some people get the vaccine and then some didn't. However, because majority of people get vaccine and they're immune to the illness and nobody else is having this illness, so this pathogen cannot pass on to get more infection. So we cut the circle of spreading. When majority of people get the vaccine and a small amount of people didn't get vaccine, and we prevent the spreading of the disease, then this group of population is immune to this bacteria, for example. It's called herd immunity. But be careful, tetanus is from the rusty irons, I think. It's impossible to eradicate. It's not like a live bacteria. So we cannot have herd immunity for tetanus. That's it for lecture 5. Let me see lecture 6. Okay, different communications. Pathogen communication. How pathogen is communicated is they secrete chemicals to detect surrounding population. When they have a large amount of population, then they will produce toxin together. That makes to harm the object effectively. Multicellular communication is, for example, the ghrelin secretes, the stomach secretes ghrelin and then stimulate hunger, like the communication between cell and cell. It can use hormones. Direct signaling, like, for example, the cardiac cells, they're sticking together and they're able to directly communicate between cells. And cell-to-cell communication, there's distance between each cells. So, for example, but they're close enough so that they are able to bind to each other. For example, APC and the T cell, T helper cell. Chemical signaling target cell ligand is when there's neurotransmitter is an example. It's between the neurons. Autocrine signaling is when a cell secretes a hormone and this exact cell has the receptor and this hormone can bind to its receptor on itself. So it's This is the meaning of this term. Autocrine, just communicating with itself. Paracrine signaling. So it's about excreting chemicals to a neighbor cell. Para means near. Endocrine signal means the signal is secreted into the blood. We're using blood vessel as a passage way to communicate. So the difference between neurotransmitter and neurohormone, whenever there is a blood vessel that's involved, makes it neurohormone. Hydrophilic vs hydrophobic hormone. So phobic is afraid, hydro means water. So something that's afraid of water is hydrophobic hormone. Why do we need to know this? It's because different types of hormone enters the cell membrane differently. Remember, the cell membrane is made by phospholipid. It's a fat. So hydrophobic cannot cross over. Only hydrophilic able to cross. So if a hydrophilic is able to cross the membrane directly, then it's fast. And then it has a strong effect. However, it comes and goes fast. So it can hydrophilic. Oh, yeah. Hydrophilic is the water type. Water type likes, philic means like, so like water. It's a water made by water hormone. The water cannot enter the phospholipid directly, so it's stopped by the membrane. However, there's receptor on the membrane, and then once it binds, the hydrophilic hormone binds to the receptor on the membrane, then it signals something inside and then cause a cascade. So that's why it's an amplified effect. And example can be insulin, epinephrine. And hydrophobic is made by fat, afraid of water. So it's able to go into the cell membrane and it lasts longer. It's hard to get around in the bloodstream. An example can be steroid hormone. There's no need of receptor. It's able to cross the cell membrane directly. But it has a less stronger effect than less effective effect than the hydrophilic. Okay, so now we're going to talk about what happens if it's hydrophilic hormone. Hydrophilic hormone made by water cannot cross the phospholipid bilayer. It will bind to a G receptor, bind to a G protein. There's receptor here. And then under the G protein, we have GDP. So with the hormone bind to the receptor, it signals G protein to have drop GDP, and GDP pick up GTP. GTP will, no, this one is, no, let me talk about this. So once it binds, the DP will become GTP, and GTP just leaves the G protein and bind to an enzyme. For the test purpose, you don't need to know which enzyme it is. You don't need to memorize this name. So GTP binds to an enzyme that signals ATP to become CAMP. So CAMP is like a second messenger. It's going to go into the cell and signal other proteins and eventually into the nucleus to make some change. So depends on where it goes into. If it's only the effect is taking result in the cytoplasm, then it's called cytoplastic response. If the cascade goes into nucleus, it's called nucleus response. Usually, nucleus response is produced by hydrophobic, the fat hormone. That will change the DNA and then produce protein. Here is talking about the same hormone, but it has it has multiple receptors. Depends on which receptor it binds to, it produces different effects. For example, epinephrine. If epinephrine is bind to liver, then that will cause glycogenolysis. And if it's at the smooth muscle, there will be vasoconstriction. And if there's in the cellular, it increases cellular respiration at the skeletal muscle, and it increases respiration rate. It inhibits the digest system because it's the parasympathetic. It has inhibitory effect on bronchial to relax and dilate bronchial. That's it. See you in next video. Fighting for exam. You got this.